Hi, I'm Amanda.
I'm Jake.
I'm Jill.
And I'm Prima.
So migraines affect 14.7% of the global population, which makes it the third most common disease in the world. Besides this prevalence, it's also the seventh most debilitating condition, causing 2.9% loss of years of life. Some patients have chronic migraines, which are characterized by headache episodes occurring more than 15 days in the month. And this affects 1.4 to 2.2% of the global population. With such high prevalence and debilitation, this disease requires solutions to improve the patient quality of life. The main routes to alleviate symptoms are through two kinds of medication, acute and preventative. Acute medication is often used to relieve symptoms near the onset of the headache, where preventative medication is taken to reduce the intensity, length, and frequency of migraine episodes. Preventative medication is generally used when warranted by frequent and severe migraines, but over 90% of all migraine sufferers use acute medication to relieve their symptoms. Some clinical evidence suggests that using acute medication before the onset of symptoms improves the treatment efficacy. Taking medication before the headache starts may also reduce the prevalence of over-medication. So how do patients medicate early? Well, around 1/3 of migraine patients experience what is called an aura, which often manifests as visual or sensory disturbances that precede the onset of a headache. This can be used as a warning system, and some evidence suggests that medicating during the aura phase can prevent headache onset in over 80% of patients. However, this does little to help with the 2/3 of patients who don't have auras. And for those who do have auras, as high as 81% of them also have migraines without auras. So while some patients do have an aura that will allow them to have that warning system, there's a need for a device that warns all patients when a migraine is coming on so they can medicate and improve their quality of life.
So it is important to understand the pain pathways because a lot of the current therapies will work to target this. And the red boxes show the 3 different parts of this pain pathway. So the first phase is that the stimulus will reach the different touch receptors, and this will activate the pain or nociceptors, and they'll detect pain as part of that pain pathway. So this is going to be part of the peripheral nervous system, and then the signal will be transferred to the second part, which is the dorsal root ganglion. And this helps to connect the peripheral nervous system to the central nervous system. So then the dorsal root ganglion will transfer the signal along the spinothalamic tract to the cerebral cortex in the brain. Once it reaches there, the signal will be reached by the thalamus, and the thalamus will then relay that signal coming in from the senses to the sensory cortex. And after the signal has been processed and understood, then an appropriate response can be generated down the descending pathway via the reflex organs. So the most important part of the pain pathway is the dorsal root ganglion, which is where this red arrow is pointing, because a lot of therapies work to target the nerves that are part of that complex. So currently on the market, there's acute or preventative drug therapies, and the acute medications are usually taken during a migraine attack, and then the preventative medications are taken in order to prevent the migraine attack. So acute medications include the common over-the-counter medications like Advil, Tylenol, or ibuprofen, and preventative medications include a class of drugs called triptans. So triptans, as you can see in the left picture, work to inhibit the exocytosis of neurotransmitters from the trigeminal nerve. Specifically, these neurotransmitters are CGRP, NKA, and substance P, and these neurotransmitters are involved in nociceptive or inflammatory responses. So trying to inhibit the release of these neurotransmitters will help to reduce the migraine pain. A common problem with a lot of the medications is side effects. We also have stimulation devices on the market. The left shows an image of the non-invasive vagus nerve stimulation. So as part of this device, we have the device that's outside of the neck, and it works to stimulate the vagus nerve. And stimulating this nerve, which is part of the pain pathway, helps to reduce the migraine pain. We have the sphenoganglion stimulator implant on the right, where we have a controller that works to activate the implant on the inside of the mouth. And then that implant will go on to stimulate the SPG nerve bundle, which also helps to alleviate migraine pain. However, this has only been approved in Europe. So the problem and limitations of the current therapies on market is that there's a lack of diagnostic treatments and there's no continuous monitoring for patients. So we're trying to have a diagnostic treatment so that the patient can be informed of the migraine before it even happens. And the continuous monitoring for the patients is really important as well, because each patient will have different chemical levels and they'll respond differently to treatments. So our device is trying to target these areas and bring additional value to the market.
We propose that an implant that monitors the concentration of neuropeptides that are linked to the onset of migraines could alert patients to oncoming migraine attacks days earlier than self-monitoring symptoms alone. The neuropeptide that we will monitor is calcitonin gene-related peptide, or CGRP. Several recent studies have indicated that CGRP levels in the saliva increase during a migraine and go back to normal after a migraine ends. It's important to note that what counts as normal varies for each unique patient. Our device will initiate a trial period in the weeks following initial implantation. The device will begin to monitor CGRP levels while the patient logs migraine incidents into a mobile app. Once the device learns how much CGRP is typically present in the saliva directly before a migraine, the device will switch to active mode and begin to warn patients of oncoming attacks via the same mobile app. The implant will be placed in the cheek tissue near the parotid salivary gland and its associated Stensen's duct. On the screen, you can see a diagram of the salivary glands in the mouth. The proposed location of the implant is boxed with the black square. The base component of the implant would be composed of PDMS, a silicone elastomer that is nearly bioinert and does not elicit a negative immune response in the mouth. The modulus of PDMS closely matches the modulus of cheek tissue, limiting patient discomfort. The base would be manufactured by mixing a curing agent and siloxane in a custom ratio to achieve desired stiffness. The PDMS would be degassed in a vacuum chamber and poured into a custom negative mold. After the PDMS was fully set, it would be removed from the mold. The PDMS would be sterilized by immersion in 70% isopropyl alcohol. Antibodies that are specific to CGRP will be conjugated to the surface of the PDMS base component to measure CGRP levels. To limit biofouling and nonspecific binding that could cause sensor blindness, the device will be coated with a thin layer of poly-L-lysine PEG graft to discourage nonspecific binding and biofouling from proteins in the body. The PEG polycation coating would be applied by treating the PDMS surface with oxygen plasma. The polycationic PLL backbone of the molecule is attracted to the negatively charged surface, leaving the PEG chains exposed to the environment. The antibodies will be conjugated to the surface using covalent bonds. The PEG would act as a long spacer that allows bonds to form between its hydrophilic matrix network and the antibodies. You can see in the diagram in part C that the protein is conjugated to the surface with a covalent bond, and you can see in part D how a spacer might work. The base component of the implant will be sewn into the cheek tissue using non-resorbable Gore-Tex sutures. Sutures can be chosen based on the mechanical strength necessary for a specific patient. The porous nature of the sutures allow for tissue attachment, and the nearly inert nature of the threads would dramatically decrease the risk of a negative tissue response to the attachments. Gore-Tex sutures are already sterile when they're removed from the package. The base component of the implant would be surrounded by a porous casing to limit the exposure of the antibodies to bacteria in the mouth. The casing would be composed of a polyurethane matrix with silver nanoparticles embedded throughout. These nanoparticles have been found to inhibit the activity of both E. coli and S. aureus pathogens in vitro with low toxicity to human tissue. The polyurethane casing would also have a modulus that resembled that of PDMS. This casing would be synthesized by adding silver nitrate and tannic acid to a polyurethane solution prior to the electrospinning process, allowing for an even distribution of the nanoparticles. To measure the levels of CGRP that bind to the antibodies, the device would utilize a semiconductor electronic label-free assay, or SELFA, that uses electronics to quantitatively measure ELISA results by quantifying the concentration of antibody-antigen conjugates in a system. The SELFA can be connected to portable readout electronics to share results and store data. The system also demonstrates similar selectivity to ELISA assays, which limits the risk of measuring the concentration of molecules other than CGRP that don't tell us what we want to know. Patients will rinse with a PBS mouthwash 2 times per day. This mouthwash will act as a wash buffer that clears the CGRP molecules from the antibodies while leaving the antibodies conjugated to the PDMS PEG base component. PBS is already sold as ELISA wash buffer, and the safety data sheet for the buffer notes that the substance is not toxic, nor does it contain toxic substances. When the patients rinse with the mouthwash, they will reset the device and allow for monitoring of oncoming migraine risk with the app. This portion of the device could be sterilized using vaporized hydrogen peroxide sterilization. While this device could be beneficial for a large population of patients, there are potential drawbacks. The first is that the device would be needed to be replaced once per year to ensure that the sensors remain sensing CGRP accurately. However, some patients might not want to have even a minor surgery this often. Next, the device does not actually provide treatment once it detects a migraine. Instead, it just warns the patient, so the patient is responsible for taking medication. If the patient does not follow through on medication, then they will not feel the effects of less pain in the migraine. Finally, the data that is shared between the app and the implant might be corrupted by hackers or people that want to see the data other than the patient and the healthcare providers. This could pose a risk to patient safety.
In order for the device to be determined as successful, it must maintain basic functional requirements when implanted into the body. The functional requirements of our device include that the device must read the CGRP levels in the saliva of the patient on a daily basis. The device must be able to determine when the CGRP levels are higher than normal. The device must be able to understand the patient's baseline levels of CGRP. The device must be able to alert the patient of an oncoming migraine. The sutures of the device must maintain the device's placement and negate any migration. And lastly, the wash buffer with the device must clear the CGRP molecules from the antibodies. Along with this baseline functional requirement of the device must also hit a specific design benchmark for the device to be considered as successful. These include the ability of the device to be biocompatible once implanted. The device must discourage biofouling in the mouth and specifically around the implantation area. The device must last a year once it has been implanted in the body while maintaining the same level of biocompatibility and without any signs of degradation. The device must be relatively inexpensive and easy to manufacture. And lastly, the device must be comfortable to the patient and almost seem non-existent after implantation. There are also multiple expected mechanical properties for the device to be able to be functional within the cheek while maintaining comfort within the patient. And these properties will include the Young's modulus, the tensile strength, the maximum load, and the thermal conductivity of the device, as well as the mechanical tensile strength of the sutures themselves. The device will also include 5 main materials that will interact with the tissues in the cheek as described in the proposed approach. The first material is PDMS, which will, when implanted into the body, is proven to be bioinert, non-toxic, and biocompatible. The second material includes PEG, or polyethylene glycol, which will be used as a coating and interacts directly with the cheek tissues. PEG is widely used in biochemical and medical applications and is highly soluble and biocompatible. Polyurethane, which will be used with silver nanoparticles, will act as an antibacterial medium and has low toxicity and has been used in multiple medical implants that interact with tissues in the body. The Gore-Tex sutures being used are made from polytetrafluoroethylene, which is a non-degradable porous polymer and is non-adhesive to tissues and is hydrophobic and bioinert. And lastly, the mouth will be washed periodically with PBS, which is non-toxic and commonly used in vivo and is a non-binding material.
So we will perform mechanical testing on all of these materials in tensile, compressive, and torsional conditions as laid out by the ASTM standards. And this will output modulus strength, fatigue resistance, as Jill mentioned earlier. And we will use these values to compare to both the theoretical value for the material we should have, as well as the properties of the cheek that are required. Some limitations are that these properties may be different in vivo due to water plasticization, so we will address that as well.
We will also be performing multiple biocompatibility tests within the device. These will include a leachable and extractable test, which will involve placing the components and materials of the device in an extraction media to evaluate any of the leachable components that come out of the device for any cytotoxicity. And we'll also be completing a systematic in vivo toxicity test, which will determine if any of the materials we will be using contain any chemicals that will cause negative effects after any prolonged exposure.
We will also perform a test to evaluate the specificity of the sulfa that will be used to evaluate the concentration of CGRP in the saliva. The research team would obtain a set of 15 solutions that possess 5 different known concentrations of CGRP, 5 solutions that have no neuropeptides at all as a control, and 5 solutions that have molecules that are biologically similar to CGRP in order to stress the specificity of the system. The researchers would use the sulfa and ELISA kits from at least 3 different companies to evaluate the concentration of CGRP in the sample. These results will be compiled and subjected to statistical analysis using non-parametric tests such as the Wilcox and ring sum test. If the values obtained from the kits are not significantly different for the sulfa than the values for the known concentration from the ELISA kits when alpha equals 0.05, the sulfa will be deemed acceptable for use in the device. This test is limited in that there is no setup in which CGRP is mixed with other molecules that are biologically similar to compete to bind to the receptor. This would very likely be the case in the saliva, so future testing would likely need to be performed.
We'll complete additional testing on the antibacterial properties of the device, biofouling, sensitization, and hemocompatibility to make sure it doesn't elicit any unwanted responses, and we would do characterization testing to determine how long the device would last, whether the device would migrate from its original position, and to figure out the optimal pore size for the CGRP molecule. Thank you for listening to our presentation.